Welcome back. Alrighty. So before I dive too deep into the next little section, I did want to ask you guys how you feel about the YouTube channel, because I don't plan to make money on it, but I did want to, for future reference, I wanted to see how you guys felt about me uploading recordings there, so they're a little bit more accessible. Like you could look it up on your phone, on your computer. It doesn't matter really where you are. You could probably see it. And I was also thinking about, maybe instead of doing Zoom sessions and then having to upload them, I could just go live on YouTube, and then you could get the notification. You could get and then automatically upload my lives. Like, what do you guys think about that? The whole YouTube ideas. I just want a little bit of Thank you. Honestly, I think it would be a little bit less work because for Zoom, I of course have to log on, but it doesn't automatically save it or anything. Like I have to go in and do that, and then I have to save it to my computer, and then after I save it to my computer, only then can I make it a shareable link with you guys, and then I have to put that in Remind. And I know it gets to be a lot of notifications, but I think the accessibility aspect, because you can only get the link if you have Remind, so to the recordings at least. So my idea was, if I give it to you on YouTube, then you could do it on your computer. If you don't have Remind on your computer, you could get it on your phone if you really don't want to download Remind. But yeah, and then also I think it's a little bit easier to to pause and stuff. Plus, I plan on YouTube to upload not just recordings, but also like review videos. So like, if you guys wanted me to like specifically go over TSIs, like I plan on doing that probably either today or tomorrow. But I'm gonna make a TSI review video because it's one of those things that like falls by the wayside. It's on exam 3, but you did it before exam 2. So I'm planning on doing like a review video like that, or different other ones. But like, I think that it might be a little bit better than the current situation I have right now. So yeah, but let's go ahead and dive back in to epidemiology. So let me share my screen so that I can get you guys up to speed. Woohoo. And why is there a forehead on my slides? Okay, there we go. For this section, it's mainly about the whole progression, right? So this is the biggest takeaway. This screen, you have your portal of entry, the adherence, and the number of invading microbes contribute to its penetration or evasion of host defenses. And then you have damage, and then you have portals of exit. So if you can take a disease that you know a lot about and say, okay, this is a portal of entry, this is what it contributes to penetration via these two things, and then what is its special characteristics that penetrate or evade host defenses. How does it damage the cell, and then how does it exit the body? So a lot of like GI diseases, obviously the portal of exit is going to be your feces. So, and more than likely the portal entry is going to be through your mouth or through ingestion. That's more scientific words. So technically that would be a mucous membrane. So your respiratory tract infections, GI, genitourinary, and conjunctiva, right? Pink eye, you get it through your eye, and you can secrete it through your tears, right? And it has a slew of different characteristics. So go through these and understand what the bolded words mean. Like if it's bold, then most likely need to know it. It's pretty gnarly, huh? Thought that was pretty cool. Let's see. So also another thing, this should be bolded. Not all portals of entry are equivalent. So I think one like key thing that you guys should remember is that, while E. coli in your intestines are fine, if that E. coli gets somewhere else, that's when it becomes a problem. So sometimes it's, the bacteria on your body gets in the wrong place, and then you develop an infection. So, we talk about all these bacterial agents and how ingested they can cause this. Oh, but also they live on your skin. How does that make sense, right? So another thing is like, UTIs. That's by not wiping the correct way, right? That takes our normal flora and puts it somewhere it shouldn't be. And that causes an infection. So this is the other side of it, is that if it doesn't enter in its preferred way, right, then it's not going to cause a disease. So similarly to salmonella, if it is not ingested or it is not through a mucous membrane, E. coli will probably not cause you harm unless you have an open cut on your hand. Then it'll probably cause harm. So if it just sits on your skin, we have the protective layers that we can wash it off and we'll be just fine. But if we swallow it, there's no way to really fix that. Or if we get into a cut, we need to really, really thoroughly rinse that. We need to get that out. But some microbes do prefer a certain way and will not cause disease any other way. This is usually, this is usually bacteria that I want to say, I just lost my train of thought. I am sorry. We'll see some examples in a second. Ulu. I don't know if this is going to be on the quiz, but it's definitely going to be on the exam. So ID 50 versus LD 50. So ID is the dose required to infect 50% of the population. LD 50 is the potency of toxin that is required to kill 50% of the population. So remember, infection with ID and LD, just remember it's the death one. Like it's kind of hard to remember because like what is L supposed to stand for with this one? Oh, I'm stupid. It's supposed to stand for lethal. So it's infectious dose versus lethal dose. My bad. But remember those two because people confuse them sometimes. And that's pretty much it. Also, I think we also talked about this, the difference between bacterial loads. So if you have a lot of bacteria and you slap that on an open wound, you're probably going to get an infection. But if you take one bacteria and like smidge that into an open wound, it's a variable whether or not you'll get an infection from it. Let's see. This is the very, very blatant example, is that, anthrax, it's very, very infectious on the skin, but if you inhale it or if you ingest it, it's not as infectious. This one likes certain portals of entry. This is what I was talking about. And remember, ID 50 gives us how its preferred portal of entry. So if its ID 50 is, let's say it's for inhalation, it's 1 to 50. For skin, it's the 10,000 to 20,000. And then ingestion is still the same. Then we know that its preferred portal of entry is going to have less bacterial load needed. Okay. And then look at this. Botulism is a scary, scary, scary disease. Because look at that. Nanograms per kilogram. Most of us are only like, I want to say anywhere from like 65 to 100 kilograms, like most of us. I know that's kind of being a little bit on the shorter end, but you get what I mean. So per kilogram, that's not a whole lot. For nanograms are always really, really small. So you can see how much this is the LD. This is how much kills you. This is scary. So look, it's about 8,000 times more lethal than Shiga. And botulism, you guys will learn later on, but it's from the canning industry. It's kind of, like you won't look at a can the same, but botulism is a disease cause, and it's usually from canning, while the Shiga toxin is usually from the specific serovar for E. coli. It's like OH7 something or other. You know what I mean. It's that weird one that has a bunch of numbers and letters and in H somewhere in there. That has the Shiga toxin, and that's from Shigella. So if you get Shigella dysentery or hemorrhagic colitis or that specific E. coli infection, that's how much of the Shiga toxin it takes to kill you. And it takes a lot of bacteria to produce that much Shiga toxin. So you'll most likely vomit all that toxin up before it actually hurts you. Oh no, I'm sorry. Gosh. Okay, so how far, you guys didn't go over this at all? Okay, well, I guess that would have been a good, you're fine to interrupt. This is important. Okay, well, you guys get a little preface, 13 slides preface. So, I'm sorry, but yes, that's your little preface to this one. It's a little bit more about epidemiology, a little bit deeper. So let me just switch over to, I would think that she would do, well, I guess she would have done, because I feel like for my lab, we did the quiz on both. So I don't know if I was just making that up, but we're switching gears to viral structure. So you guys saw from your last quiz, that it was like the first half of this, right? So your viral structure, make sure you know that for your exam, capsule versus capsomere. But I was told that it's probably the latter half. So before your guys' quiz, last quiz, what was the latest, like what was the middle portion that she went over? Because I went to the lecture, but I can't quite remember exactly where she stopped. In terms of the material that was on the last quiz versus the material that's going to be on this quiz. I want to say mid 13. Okay. Okay, so it's 69. So let's start here. This is good. So you want to know your differences between DNA and RNA viruses, single-stranded versus plus sense, minus sense, double-stranded, and retroviruses. There aren't a whole lot of double-stranded RNA viruses, but there are still a few. Let's see. So here is your one exception you need to memorize, where the pox viruses, where those replicate. This is a good one. So a lot of the times if a virus has a, or I'm sorry, a lot of times when the virus is coded, right, it taps into the bacterial cell wall, and then it likes to merge with it, and then it kind of sneaks its way in and gives it its DNA, and then so like the cell's like, okay, I'll replicate this, and then it creates so many more that they just keep going and infecting cells, and that is usually how they replicate, because remember, viruses aren't considered living. They do not have their own ribosomes, and they are not capable of undergoing protein synthesis. So they are unable to grow and reproduce on their own. They need a host always. Let's see. Entirely in this cytoplasm. Okay, so one thing with the minus sense is that it has to be flipped. So the plus sense can go in, do its job, replicate, it's fine. But the antisense or the minus sense RNA has to be translated. So it has to come with the RNA polymerase so that it can be translated. That is another thing, is that that's going to be in the viral package. And when we take that and we put it into a cell, it's, you know, we have our strand, it's going to be our antisense, and then we're going to have that RNA polymerase so that it can be translated and give us the essentially the mRNA that is going to be able to code for all of the viral DNA. The plus sense RNA can just go straight to reproduction. It doesn't have that much step. I don't know why nature decided to say, okay, we're going to make an antisense RNA. Like, I don't know. Like, that seems like a lot of steps to me. Why not just be a plus sense? Understand that this is a process called budding, right? They are taking part of the cell wall so that they aren't seen as foreign, and they're taking that viral DNA and going into it and then closing off, and then it becomes its own separate thing, right? That's budding. Sorry. Yes, budding. Let's see. So these are your steps. These are your, this is the reproductive cycle. This I would highly recommend drawing out. Oh, she said HIV was going to be a definite topic. So remember, I know it's like memorization galore, but memorize the what is going on here. So HIV infects T helper cells and macrophages. Okay, HIV infects both T helper cells and macrophages. You need to know that the CD4 is where GP120 connects. I feel like there is a really good video on this. I will give you guys a video that walks through this and it shows you exactly what is happening. This is, you know, it shows you the GP120, the GP41 attaching to the CD4. And the CXCR, yeah, it connects to both. And then, let's see. And then, so those are your T helpers. Then if you look at your macrophages, it connects to the CD4 and the CCR5. So T cells are CXCR4 and the CD4 molecule, while the macrophages are your CCR5 and your CD4 molecule. So there's this really weird thing that happens. So there is a CCR5 mutation that gives people a like edge to fight to not getting HIV. You're not, you're not like immune to it, but you're less susceptible. So somebody who has the complete gene is 85% susceptible. Somebody who has a partial mutation is susceptible, but it doesn't progress as rapidly. But if somebody has mutations of both CCR5s, then you have like an edge. For whatever reason, you have an edge. So this is the microscopic view of HIV viron. And for this specific virus, it can either remain latent, cause the release of new viruses very slowly, or it can just completely destroy. Usually it happens in that order, where it kind of just sits there for a little bit, make sure that it's the right conditions, and then it starts to release a little bit, test the water a little bit more, and then it just kills everything. Usually that's why you see how they start off kind of good, right? Like the infection, like if I'm looking at this graph, right? The infection starts here, right? And it kind of dips down a little bit, but it's generally pretty straight. It remains latent, right? And then it starts to kind of go down, right? It's releasing a little bit at a time. And then all of a sudden, bam, it drops significantly. So this is your CD4 cells that's being counted, right? It's destroying these cells. You can also see that I'm sorry. You can also see that in the second graph. I know it's a little bit busy, but the graph on the right, in the red line, it shows what the graph on the left is. And then in the middle, it shows the antibodies, right? Your body is trying really hard to fight this off, and then it just can't anymore. And then the virus titer, it just means the number of viruses in your body. So this is why it's like, oh, HIV, you have this. That's why a lot of people see it as like, oh, I have this infection, right? And it's like, oh, well, it went away. And then later on in life, all of a sudden you get full-blown AIDS. Like, that is a really difficult thing about this, is that you might have HIV here. It becomes difficult to detect after a while. The symptoms might go away. And then all of a sudden, AIDS. That is the infection. And usually you can see it as like, this is like the latent, this is the latent, it says, and then it starts dropping a little bit, and then you could see the viral titer coming in, and then it keeps decreasing, but your antibodies kick in here, and it almost deflects some of that virus, but the virus can stay in you for so long. That's the thing with this disease. So the antibodies kind of cancel it out here, but as soon as there's such a significant drop here, you just have AIDS here. So one thing you need to know for the quiz and the exam is, what is the definition? That is the clinical definition of AIDS. And it's right here on the slide. It's less than 200 T helper cells. So when we pass, right, this is when we have AIDS. This is 200 T helpers. This is when AIDS occurs. So just remember that I don't think you'll necessarily need to know about the antibodies, but one thing's for sure is that you'll see the latency, you'll see the slow progression, and then you'll see the drop off. And then once you pass that 200 T helpers, you are SOL and you have AIDS. I am sorry, my friend, but you would have AIDS. Another part of this is opportunistic infections, right? Those secondary infections. It is very possible that while you don't have T helper cells, riddle me this, Batman. What do T helper cells do in your body? What is the function of T helper cells? What type of cell are they? I'm looking for a chat. No chatty Cathy's today. Yeah. You guys remember AMP, right? What are T helper cells? I know I'm a little bit over. Yes, they are part of your immune system, right? They are immune cells. So if we're knocking out a big population of our immune system that helps us fight infections, what are we going to be left with? A very weakened immune system, right? So if we have this severely weakened immune system, we have the severely weakened immune system. We are so susceptible to other infections. It doesn't matter what it is. We can get so many other infections, and that's why usually AIDS patients die pretty quickly. They basically just get taken over. They no longer have a functioning immune system, and there is no known cure as of right now. You don't need to know the specifics on here. Just know that it's very likely for an AIDS patient to, and not only are they knocking out their T helper cells, but their macrophages. That's one of your secondary lines, right? It eats those foreign objects. So if all of a sudden we don't have those eaters, what's going to happen? Scary to think about. Let's see. These might look a little different on my end than your guys' ones that you went over in class, but let's see. So know this: when animal viruses become integrated in host cells, it's a provirus. And that provirus condition is permanent. It becomes part of the host's chromosome, just like HIV and eventually the full-blown AIDS. Let's see. This is a good one. This right here, the persistent infection latent type, that's like your herpes, right? God, don't we love herpes? If you get a cold sore, right, your immune system can usually keep it at bay. You get stressed, your immune system becomes, weakened. That wall falls and it lets that virus show. Okay. Remember, varicella zoster is just chickenpox. Usually, if you're a kid, you get chickenpox, but I never got chickenpox. Fun fact. Only one of my sisters did. And she grew up in the 90s. Let's see. Yeah, there is the chickenpox vaccine now. Used to that parents would just have like these pox parties is what they called them, If one kid had chickenpox, you throw all the other kids in the room so that they all get it now and they don't get it later. Because when you get it later, it's a hell of a lot worse. So, yes. Let's see. An immunosuppression right here, I was talking about being stressed and your immune system becomes suppressed. That's the herpes. That's if you have it, if you had gotten it already. Like my sister, she gets cold sores whenever she's stressed. I have had the blessing of never getting a cold sore in my life. I'm the lucky few, but cold sores, that's a type of herpes. Hate to break it to you. But when you get stressed, it resurfaces. That's because of immunosuppression. Let's see. I don't know if you guys will need to know all this. I think it's just kind of good to know. I think some of the stuff is interesting. Also, this is why we wear masks when we have something that spreads similar to a flu. Because this is what happens if you cough or you sneeze. All of that out. This is keep the mask on if you cough or sneeze. Because it just stops that. It stops a lot of it. Let's see. You will need to know the H versus N spikes. That's how we identify them. And their combinations and what the H versus N stand for. Antigenic shifts are scary. Usually they have a mixing vessel. So like, I'm pretty sure this example is the bird flu. So what happened was the human flu virus and the bird flu virus went into the mixing vessel, which is the pig. The pig then basically housed a breeding ground for these. And then the two viruses became one and incorporated just the best qualities of both. And then once that mixing vessel let that release, and now a human got it. Now we don't know how to fight it off, and we become severely become severely ill, right? So there is that. That's genetic recombination. We did that in class, right? But this is just happening. This is how it happens naturally. It's scary that these things can happen. And that's why we had a whole bird flu pandemic, and yeah, and like, I mean, yeah, I'll say that. But yeah, it's referred to as a mixing vessel. One other thing to know is that most of these, non-infectious, they just, they get their DNA, they share it, and then it winds up not infected. But those that are successful, it becomes very, very bad. It becomes a pandemic, or because like we have our antibodies from previous infections, but if we get reinfected by a completely only 50% like flu and 50% bird flu, if we get infected with something like that, our antibodies won't understand what that is, and we get the full-blown infection, and we get pass it to others. Yeah, but just know the mixing vessel as well. I think that's just about it. I'm sorry I held you guys so far over, but this is all possible stuff that could be on the quiz. But for sure, understand this. Like, I'm pretty sure I missed these questions. But yeah, I will also be, I'll send it on Remind, and I'll also send it on, or I'll also post it on YouTube as under a playlist that you guys can view. I will post a video that walks you through how HIV functions in the body and how it attacks cells, so that you guys can visualize it. It's not just a drawing on a page. So yeah, other than that, you guys are free. If you guys have any other feedback, I'll be here. Just let me know if you don't want to call me out publicly. But if you do want to call me out publicly, I'm fine with that. Um, just let me know like how I'm doing, because if I'm going to be honest, last week nobody came to my sessions. A couple people stopped by, and that was like it for like the week. And like I don't blame you if you're busy, but like I need to know what I'm doing, what I could be doing to entice you guys to come and make it appealing. But other than that, you guys are free to go. Um, no, you're good. Like I said, if you're busy, you're fine. It's just like, there's like 30 to 40 people, and out of those 30 to 40 people, 3 come, and like I appreciate you guys for coming. It does take time out of your day, and I appreciate your valuable time. Um, I just need to know, like, if people aren't doing anything, why would they not come? Especially if they have a quiz. But I also heard that people are enjoying the recordings. So I have a little bit of hope on that end. But again, if you guys are just like, can you try doing something like this? Shoot me a remind text. I don't mind trying new things. Because it's not only going to help you guys, it's also going to help anybody else that I pal for in the future. So just let me know how I can make these a little bit more enticing. And yeah, but other than that, you guys are good to go. I'm not holding you hostage. But don't get burned out. Take time to take a deep breath. Sleep. You guys need sleep. Make sure that you guys make that to-do list and kind of get crack-a-lacking. You guys are gonna be fine. I promise. It's not. There is light at the end of the tunnel, and it's closer than you think. Okay. But if you guys are good, no problem. If you guys are good, I'm good, and I will hopefully see you guys Wednesday, if not Thursday. Okay. Bye.